In this film, we delve into topics such as genetics, its history, classifications within the field, population genetics, molecular genetics, clinical, behavioral, plant, animal breeding, medical, quantitative genetics, gene linkage, gene mutations, and the relationship between genetics and diseases. Stay with us. 1. Genetics, French, génétique, from the Greek root gamma epsiloni epsilon tau iota kappa, meaning origin, birth, race, or type, also known as gene science, is a branch of biology that deals with heredity and the variations among living organisms. By applying the rules and concepts of this science, we can understand the similarities or differences between two living beings and learn how and why such similarities or differences arise within plant or animal communities. Genetics is defined as the transfer of biological data from one cell to another, or from parents to offspring and future generations. It is concerned with the mechanisms of these transmissions that result in traits, transformations, and similarities among organisms. Genetics also studies the physical and chemical nature of this biological data. 2. History. Although biology is one of the oldest sciences known to humanity, about a century ago, a new branch emerged from it, genetics, which revolutionized the field. In the 18th century, some researchers began exploring how certain traits and characteristics are passed from one generation to the next. These are referred to as hereditary traits. However, due to two major issues selecting inappropriate traits and a lack of sufficient mathematical knowledge, they failed to reach definitive conclusions. The first person to identify the rules governing the inheritance of traits was an Austrian monk named Gregor Mendel, who in 1865 presented the laws he discovered through his experiments on pea plants. Unfortunately, the scientific community of the time largely ignored his discoveries, and his work was forgotten. In 1900, the rediscovery of the same laws by De Vries, Schermach, and Correns brought Mendel's ideas back into serious consideration. Today, Mendel is known as the father of genetics. In 1953, with the discovery of the structure of genetic material by James Watson and Francis Crick, a new field emerged in biology known as molecular biology. About a century after Mendel's discoveries, during the years 1971 and 1973, the fields of molecular biology, which studied the structure and function of genes, and genetics, which focused on genetic diseases and their treatment, merged to form genetic engineering. This new discipline quickly became influential in many fields such as medicine, industry, and agriculture. Genetic research has provided humanity with greater insight into the surrounding world and a new understanding of itself. All of our physical traits, as well as those of all living beings on Earth, are influenced by the DNA in their cells or by genetic changes that occur either randomly or due to external factors within the genome. These changes usually involve the replacement of one or more nucleotides in the DNA sequence, which permanently alters the genetic information and can be passed on to future generations. For this reason, the application of genetics has become widespread. One major practical application of this science is in determining familial relationships, identifying individuals, and tracing human ancestry. Today, genetic identification is widely used in legal and forensic contexts. DNA fingerprinting through molecular techniques is utilized for various purposes worldwide. These techniques may include the use of molecular markers such as short tandem repeats, STRs, mitochondrial DNA, and single nucleotide polymorphisms, SNPs, on the Y chromosome and other chromosomes. Mitochondrial DNA is used for tracing maternal ancestry, while analysis of the Y chromosome reveals paternal lineage. Additionally, in ancient DNA research, particular patterns among SNPs are examined to categorize a sample into a specific group known as a haplogroup. A haplogroup is a collection of closely related haplotypes that share mutations from a common ancestor. Haplotypes are a group of SNPs within a DNA sequence that are inherited together. Mitochondrial DNA has even been used to identify members of the royal family of Nicholas II. In 1991, several sets of human remains were discovered in a mass grave in Russia, believed to belong to Nicholas II, his wife, the Tsarina, and 3 of their daughters. Despite the remains being over 70 years old, mitochondrial tDNA sequencing proved to be highly effective. A completely identical mtDNA sequence was found between the Tsarina, her 3 daughters, and Prince Philip, the Duke of Edinburgh, who was alive at the time and maternally related to the Tsarina's sister's maternal grandmother. 3. Classification of genetic science. Genetics can be divided into several branches. 3.1 Population genetics. 3.2 Molecular genetics. 3.3 Clinical genetics. 3.4 Behavioral genetics. 3.5 Plant genetics. 3.6 Animal breeding genetics. 3.7 Medical genetics. 3.8 Quantitative genetics. 3.1 Population genetics. Population genetics is the study of the distribution and changes in the frequency of alleles within populations, influenced by 4 primary evolutionary processes: natural selection, genetic drift, mutation, and gene flow. Population genetics aims to explain phenomena such as adaptation and speciation. 3.2 Molecular genetics. Molecular genetics is a branch of biology and genetics that focuses on the structure and function of genes at a molecular level. By applying techniques from both genetics and molecular biology, this field seeks to understand gene interactions and functions. Molecular genetics not only investigates how genes are passed from one generation to the next, but also aids in understanding developmental biology and genetic mutations that may lead to specific diseases. It also explores the mechanisms of gene transfer among organisms and the causes of genetic mutations. This field studies chromosomes and gene expression, providing insight into heredity, genetic diversity, and mutations important in both developmental biology and the understanding and treatment of genetic diseases. 3.3 Clinical genetics. Clinical genetics is a branch of medical genetics that focuses on the health and disease of individuals and families. Practically speaking, clinical genetics involves applying genetics in everyday medical practice. While other subfields like molecular genetics or cytogenetics are lab-based, clinical genetics deals directly with patients. Clinical geneticists are professionals with academic degrees in genetics. Unlike genetic counselors, who assist patients after a diagnosis is confirmed, clinical geneticists may also participate in diagnosing and even treating certain genetic conditions. 3.4 Behavioral genetics. Behavioral genetics is a branch of genetics that studies how genetic factors influence behavior. The central message of genetics, and particularly behavioral genetics, is genetic individuality. Except for identical twins, each of us is a unique genetic experiment, never to be repeated. This uniqueness serves as the foundation for developing a philosophy about human dignity and worth. Human variation is not a flaw in the system. Rather, genetic diversity is essential to life itself. The influence of genetic factors on complex traits such as behavior does not negate the importance of environmental factors. For simple single-gene traits, like eye color, the environment may have minimal effect. However, for complex traits, environmental influences are often just as important as genetic ones. For instance, when one of a pair of identical twins develops schizophrenia, the other does not develop it in about half the cases even though their genetic makeup is identical. Such differences are only explainable by considering environmental factors. In fact, genetic studies provide some of the strongest evidence for the role of the environment. Behavioral change in humans depends on changes in genes, which occur gradually over generations. Plant genetics is the study of genes, genetic variation, and heredity specifically in plants. While generally considered a subfield of biology and botany, it often overlaps with many other biological sciences and is deeply tied to the study of information systems in biology. In many ways, Plant genetics is similar to animal genetics, but there are key differences. The founder of genetics, Gregor Mendel, was a 19th century scientist and Augustinian monk. Mendel studied the inheritance of traits and identified patterns in how traits are passed from parents to offspring. He observed that organisms, specifically pea plants, inherited traits through discrete units of inheritance. This term, still used today, is a somewhat early and broad definition of what we now call genes. Much of Mendel's work with plants still forms the foundation of modern plant genetics. Like all known living organisms, plants use DNA to pass on their traits. While animal genetics often focuses on lineage and parentage, this can be more complicated in plants due to their ability to self-fertilize. Plants also have unique genetic characteristics, such as being highly tolerant of polyploidy, which makes speciation easier in many plant species. Plants are also unique in their ability to produce high-energy carbohydrates through photosynthesis, a process carried out by chloroplasts. Chloroplasts, like mitochondria, have their own DNA. This makes chloroplasts an additional reservoir for genes and genetic diversity and introduces an extra layer of genetic complexity not found in animals. The study of plant genetics has major economic impacts. Many crop plants are genetically modified to increase yield, improve resistance to pests and diseases, tolerate herbicides, or enhance nutritional value. 3.6. Animal breeding genetics. Animal breeding aims to improve breeds to produce animals with higher productivity and economic benefit. Through selective breeding, animals with small bodies and low productivity have been transformed into animals with larger bodies and higher performance. All efforts and activities intended to increase productivity and profitability in future generations of animals are known as genetic improvement or selective breeding. Every animal possesses a wide range of traits, color, body size, presence, or absence of horns. These are just a few of the characteristics passed from parents to offspring and are controlled by genes. Genes are tiny, complex molecules located on chromosomes, and each animal has thousands of gene pairs half inherited from the father and the other half from the mother, forming the genotype of the animal. The genotype largely determines the animal's potential performance across various traits. Each gene pair controls a set of characteristics. Some traits have high heritability, meaning they are strongly influenced by genetics, while others have low heritability, meaning environmental factors play a larger role. When a trait has high heritability, it can be improved quickly through breeding. On the other hand, traits with low heritability require many generations to show significant improvement. In simpler terms, improving such traits takes time and sustained breeding efforts to firmly establish them. For example, in dairy cattle, traits related to milk quantity and quality are typically the focus of improvement. In poultry, traits such as egg hatchability, growth rate, vitality, and overall liveliness are considered. Environmental factors like nutrition, milking methods, and others also influence whether an animal reaches its genetic potential. In truth, an animal's total performance is a combination of genotype and environment. The genotype provides the potential, while the environment provides the conditions needed to express that potential. In other words, both phenotype and genotype are crucial and determining factors. 3.7. Medical genetics. Medical genetics is a specialized branch of medicine that involves the study of heredity and genetic variation in relation to health and disease in humans. It is a subfield of human genetics, which focuses on how traits are passed from one generation to another in humans. The part of human genetics that investigates the inheritance of traits in the context of human health and disease is referred to as medical genetics. 3.8. Quantitative genetics. Quantitative genetics is a branch of population genetics that deals with phenotypes that vary continuously, such as height or body mass, as opposed to discrete, clearly defined traits controlled by a single gene, like eye color. Both fields use allele frequencies from various genes in reproducing populations, gamodemes, and combine them with Mendelian inheritance principles to analyze inheritance patterns across generations and their descendants. While population genetics may focus on individual genes and their metabolic products, quantitative genetics focuses more on observable phenotypes, offering a more summarized view of the genetic background. 4. Gene linkage. The phenomenon of gene linkage was introduced by Thomas Hunt Morgan in 1903. Morgan demonstrated that chromosomes carry hereditary factors, genes, and pointed out that the number of genes far exceeds the number of chromosomes. Therefore, each chromosome can carry multiple genes. 5. Gene mutation. A gene mutation refers to any permanent change in the structure of nucleic acids that make up the genetic material of a living organism, resulting in phenotypic changes. An organism whose phenotype has changed due to a mutation is called a mutant. Phenotype refers to the observable characteristics expressed by a gene. For example, genes that control skin color, their phenotype is the visible color of the skin. Any change to these genes can affect their expression and ultimately alter the phenotype. For example, skin color. 6. Genetics and diseases. Today, modern medicine greatly benefits from large-scale genetic research, especially in the field of cancer. Accurate classification of mutations in cancer-related genes is a crucial step in the diagnosis and treatment of these malignancies. Certain specific mutations can significantly increase the risk of both hereditary and acquired cancers. Genetic testing for both types can provide concise and meaningful insights, and with the guidance and interpretation of specialists, can play a major role in the prevention and treatment of such diseases. Support us with your subscriptions, likes, and encouraging comments so we can keep creating with even more passion and purpose. Every thought you share means the world to us.